Well, I had, in preparation for this speech, I had written the speech a couple months ago because I will also be performing the speech to my microbiology class in the end of May. So I felt like this would be a good practice for me to do my informative speech, which so happens to be my final exam for microbiology. And my topic is forensic microbiology. I watch a lot of shows like NCIS and Law and Order and all those other crime scene shows, and so I just thought this would be interesting. So, first of all, the definition of forensic microbiology is a scientific discipline dedicated to analyzing evidence for attribution purposes from a bioterrorism act, biocrime, hoax, or inadvertent microorganism or toxin release. And what distinguishes microbial forensics from molecular epidemiology? Well, first of all, we have to define what epidemiology is, and epidemiology is the study of the patterns, causes, and effects of health and disease conditions in defined populations. So now that we know the definitions of both, we can tell the difference, and the difference is that microbial forensic data must hold up not only to the scrutiny of the scientists in the healthcare community, but also the scrutiny of judges and juries. Nonetheless, work done to date on microbial epidemiology will provide invaluable starting point for the additional work that needs to be done to make microbial forensics ready for its day in court. I thought that was a really good quote from Salyers, and that's also in my bibliography at the end. So some examples that require microbial forensics. Say a gastroenterologist was accused of second-degree attempted murder of his paramour using HIV as a weapon. So basically, a surgeon purposely infected somebody with HIV. Another case would be cases of medical negligence, where a surgeon or a nurse causes a patient to contract a post-surgical infection due to inadequate hygiene. And that's kind of a big issue, especially in healthcare. And the one example that really that a lot of people do really think of with in regards to microbial forensics is the 2001 anthrax attacks. The anthrax attacks, it's basically a bacterial spore that was sent in the mail, and when people opened up their letters, they would inhale it, and obviously a foreign bacteria in your lungs or anywhere in your body isn't very good. 5 deaths occur. I'm surprised there wasn't more, but 5 deaths occur. Many became ill from it. Excuse me. So how can microbial forensics be used to track a perpetrator down? Well, you have to make sure that these spores are the spores that you are looking for. There's many different strains of it, and so how can you be so sure? And that leads me into the problems with this field. The problems, how certain is certain enough? Does it have to be 100% right? Because most of our statistics are based about 95%. So, and how will that hold up in court? Defense lawyers will be all over that, saying, well, you're not 100% sure, this isn't 100% evidence, so you can see where the problem lies there. And there's also, like I said, different strains that are very similar to each other. Collecting the specimen at the attack site, how do you know it's even an attack in the first place? And being an EMT myself, that requires a lot of training that, is beyond what I would do right now. And also, I mean, anthrax may be obvious, but maybe other diseases are not. And validity must be obtained in multiple different labs. It can't just come from one lab. Obviously, you need confirmation. However, there are benefits to this field of study. Mostly just having a well-prepared response plan may deter potential terrorists. I probably wouldn't want to go attack someplace that has a very good system that would easily find me. And also it gives people hope for when they were infected, in cases, going back to the gastroenterologist, people who are infected, it's not their fault, so it's a cool way to have that become just for other people. So moving on, an interesting study that I came across in regards to this field is assistant professor, at the University of Colorado at Boulder. His name is Noah Fehr. I'm pretty sure I'm pronouncing that name right. He conducted a study using skin bacterial communities for forensic identification. And what he did was basically somebody would type on their own keyboard on their computer. He would swab the keyboard, and he would compare the swab on the keyboard to people's normal biota on their hands. And what he found was very interesting is that more than not, the bacteria on the keyboard matched the bacteria of the owner and not anybody else. That was a big deal because it had never been done before. And also they did multiple tests with, the mice using palm bacteria, and they found the same kind of results. And they also did one that was very interesting, that they let a keyboard sit for 2 weeks, swabbed it, put it all through different temperatures and stuff, and still in all 9 cases of that study, the bacterial community on each mouse was much more similar to the owner's hand. And it points out the technique's potential use as a forensic tool is really what this is about. There are some bioethical issues, though, with this study, privacy being an issue. Going around swabbing people's keyboards is kind of invasive, if you ask me. And there was also, when you're obtaining DNA evidence, you can get fingerprints. There was a lot of legal issues with that, but how legal is getting somebody's just bacteria? So there's a fine line between what's right and what's wrong. So how can this field of study be improved? Obviously, there's always room for improvement for any kind of field. And the main one is the Scientific Working Group on Microbial Genetics and Forensics. They set a quality of standard for labs where they have to do things in a certain order, so that everyone has to do it in a certain order, and then it makes sense. And nobody's using a different technique versus this person. It's just basically to compare. So overall, I just wanted you to learn about something that I have done a little bit of just a surface study really into what we could be using and how our future in law and in biology can come about from this. And I don't know, do you have any questions at all? If you don't have any questions, I suppose then I will conclude with that, and I hope you learned something and are become excited about this as I did. So, thank you.